Yeah, I'd like to welcome everybody to the Trailblazers webinar series. The series today, or the presentation today, is going to be clinically translatable cancer research with ultrasound-guided photoacoustic imaging. My name is Drew Heinmiller. I'm the product manager of our photoacoustics products at VisualSonics. I'm just going to do a little bit of housekeeping here before we get started. So if we can go to the next slide. Just a few notes about the webinar. We're going to be doing a recording here, so if you, for some reason, you happen to miss it or drop the call or something, a recording will be available afterwards. We've got everyone's line muted for the duration of the webinar. Please use the chat window. So on the right-hand side, there's a little chat window there, and please, if you've got a question, just enter it in that chat window there, and we'll be answering questions at the end of the presentation. The presentation is going to take about 40-45 minutes, and then we'll save a little time at the end for questions. You can always go to our website, www.visualsonics.com/webinars, and check out our upcoming webinars. I'd now like to introduce our speaker today. It's a great pleasure to introduce Dr. Srivallesha Mallidi. She went to University of Texas at Austin in the biomedical engineering department, where she earned her MS and PhD degrees in 2006 and 2009. Her master's involved designing ultrasound-based multimodality imaging systems, while her doctoral work looked at the contrast agent side of things, using these systems to look at contrast agents, where she was exploring using surface plasmon resonance properties of gold nanoparticles for detection of cancer and looking at nanomolecular interactions. She's published this work in journals such as Nanoletters and Optics Express, where she's highly cited in those journals. Now, after graduation, she joined the Wellman Center for Photomedicine at MGH. She wanted to translate these imaging techniques to the clinic. She's been an NIH Ruth L. Kirstein postdoctoral fellow there, and she's currently there as an instructor at the Wellman Center for Photomedicine, and she heads projects looking at combined ultrasound and photoacoustic imaging for photodynamic therapy-based combination treatments. She's been using the VisualSonics technology for quite some time. She knows a heck of a lot about photoacoustic imaging in general, and it's my great pleasure to listen to how she's applying this exciting modality to her research, and in particular in looking at therapies and response to therapies. I'm going to hand it over now to Dr. Mallidi. Welcome.
Hello, thank you, Drew. Thank you for the introduction. Hello, everybody, and thank you for joining the webinar. It's a pleasure to be presenting here in the webinar series. So I'll get started by actually sharing my screen here. Can everybody hear me properly? Drew, can you hear me?
Yeah, I can hear you. Yeah, it sounds great.
Okay, great. Again, hello everybody. So today I'll be presenting about ultrasound-guided photoacoustic imaging for cancer research. I focus on photodynamic therapy and combination-based treatments, and how we are using these two techniques to guide us through designing different therapies for pancreatic cancer and glioblastoma. These are the two cancers that I'll be focusing today. Before going into the details of the science, I want to do a few acknowledgements. All of this work that I present is under the auspice of Dr. Taiba Hassan and her group, and funding for her lab through an R01 and a P01 program project grant, and my F32 fellowship, which is on photoacoustic image-guided therapeutics. So moving on, today we'll be talking about two topics. One is how three-dimensional ultrasound can be used to monitor tumor progression in orthotopic models, and ultrasound-guided photoacoustic imaging for monitoring treatment efficacy, and going beyond that and trying to identify tumors that do not respond to a particular treatment and have the chance of recurring at a later time point. And we would want to use the photoacoustic technology to answer those questions. So moving on to the first topic, Most of this work is going to be in pancreatic cancer, as I mentioned previously. So as most of you know, it is the leading causes of cancer. The overall median survival is less than 6 months. So the current first-line treatment is surgical resection, followed by gemcitabine, and that hasn't really improved any survival. The coming of FOLFIRINOX, which is toxic, which is a combination of 4 different chemotherapies, only partially increased the survival by 4 months. So our goal was to design combination therapies primarily based on PDT, which is the photodynamic therapy, which is a photochemistry-based technology that combines a photosensitizer and light to cause reactive oxygen species to kill tumor cells. So through seminal work by Dr. Hassan and also Stephen Bond, they have shown that in a phase 1-2 trial, the survival is about 9.5 months with PDT. And more recently, one of the components of FOLFIRINOX, which is irinotecan, a trademark drug for Merrimack Pharmaceuticals, MM398, which is liposomal formulation of irinotecan, has shown very good efficacy and enhanced survival by 2 months. So our rationale was to combine these two therapies. I'm not going to go into the mechanistic details and the biology aspect of this combination, given the focus of the of the webinar is more on imaging. But I would want to show how we used ultrasound to longitudinally monitor tumor response to these therapies. So moving on, previously, before we had the Vivo laser system in our lab, we would implant the tumors and in the pancreas through surgical procedure and do treatments on day 9, and would have to euthanize mice on several days to monitor the efficacy or the tumor growth, and only be able to look at it at one time point, the end time point, and for these different therapies. But with the availability of 3-dimensional ultrasound imaging, we were able to do longitudinal tumor volume measurements. So one thing that really helped us is we implant the tumors, let's say on day 0, and we do the treatment on day 9. Sometimes, due to several effects, the tumor could grow, or some of the tumors are really large and some of them are small. So it really helped us in organizing the treatment groups. So even though we randomize it, we don't want all like huge tumors to go into one group and all the small tumors to go into another group. So it has helped the other sizing and tumor volume measurements helped us to organize the treatment groups so that there's a good variation, and also we know which size tumors we are treating in these combination treatments. After we do the treatment, we used ultrasound imaging every 3-4 days and monitored it until we give additional therapy or euthanize the mice until when they are moribund. Or when they're really weak, sorry. So by doing this longitudinal imaging, we also reduced the number of animals that we used to really come up with good statistics and also show synergism between the combination treatments. So the slide that you see here is an ultrasound images of orthotopic pancreatic tumors before doing treatment for 4 different treatment groups. You see the no treatment group, the irinotecan group, the photodynamic therapy group or the PDT group, and the combination. And as you notice, the tumors are about 3 to 5 millimeters in diameter and are almost approximately the same size in the 4 different groups. And looking at the no treatment, if there's no treatment given to the tumor keeps growing. So 2 weeks post-treatment, the tumor is almost 6 to 8 millimeters in diameter. And with irinotecan or PDT treatment, we do see the tumor still keeps growing, but is not as huge as the no treatment group. And in the combination, which is the combination of irinotecan and photodynamic therapy, the tumor volume is significantly lower compared to other control groups. So looking at this is the images, the imaging data, but we did several mice, approximately 7 to 11 mice per group, and what we noticed is the combination treatment is the effect is more effective compared to the single treatments alone. And so previously we would only have this data point at day 30 where we would euthanize the mice, extract the tumors out. But now with ultrasound imaging, we were able to look at longitudinal tumor progression with different treatments. And this becomes very important, especially if we are trying to do secondary treatments or treat the tumor several times. To really have a better therapeutic efficacy. So we did this in another set of mice where we did not euthanize. So euthanize the mice. We did not euthanize the mice and wanted to see at what time point the tumors are actually regrowing back. So here again, I'm not going to go into details of the different groups. They're all control. What you want to focus is on the red line, which is the combination treatment, which shows the tumor volume is much lower at around 4 weeks post-treatment. And before starting the treatment, almost all the tumors are approximately at the same time. So we gave the treatment, first treatment on day 9, and around 4 weeks post-treatment, we started seeing that the tumor started regrowing back, and which is when we gave a second treatment, that is the combination therapy group received the combination treatment, the no treatment, of course, there was no treatment to that group. The other controls, the same treatment was given to them. And you can see the second treatment actually controlled the tumor progression. It almost was stalled the tumor growth, versus in other treatment groups, either initially showed some response, but then the tumor started regrowing back. So through these experiments, we were able to reduce the number of mice to be used. Also, were able to decide on time points where a second treatment could be given. Moving forward, we would want to test these combination therapies in transgenic models where ultrasound imaging will play a huge role in sizing and determining the tumor volume. What I presented is just one part of the ultrasound imaging technique. There's a white paper available at VisualSonics, which shows the applicability to different cancer models, such as breast cancer, prostate cancer. Pancreatic cancer is just one of the examples. There are other features such as Doppler and contrast imaging that can also aid in designing and monitoring the tumor progression with different therapies. So moving on to the second part of the talk, which is photoacoustic imaging for monitoring treatment efficacy. We'll start off with a broader question. So let's say we have a tumor. If no treatment is given to the tumor, which you see is the black line here, the tumor keeps growing over time. Ideally, if we give a treatment, you would want the treatment to completely cure the tumor. That is, you want the tumor to go away and no regrowth to happen or no recurrence to happen. But that's in an ideal situation. Some therapies, there are some tumors don't respond to treatment, so we do have non-responders in some case. So the first question comes in is, can we identify the treatment responders from the non-responders? That is, can we identify the blue line from the tumors that fall under the green line? And the second question comes in is, there are some tumors that sort of seem as if they're responding to the treatment, which is the red line that I show here, but eventually regrow and become much more worse than the non-responders. And often it is difficult to treat these tumors with the initial treatment that was given, And so the second question would be, can we predict this tumor relapse at an earlier time point so that they could be treated effectively instead of waiting at a much later stage when they have become very aggressive? So the goal was to answer these two questions, and we focused on blood vessels, and as all of you know, blood vessels feed the tumors, and that's where the oxygen for the tumor cells comes from. And in particular, we are focusing on looking at changes in blood oxygen saturation, which is a ratio of oxygenated hemoglobin to the sum of oxygenated and deoxygenated hemoglobin, the total hemoglobin levels. And our hypothesis was, can this change in oxygen saturation after treatment predict the treatment response and recurrence in tumors? So to do that, we looked at several optical imaging modalities. And what you see here is a graph where we have the penetration depth on x-axis and the resolution on the y-axis. So there's standard microscopy and confocal, which are very difficult to do in vivo because of lack of penetration depth. And on the other hand, there's diffuse optical tomography, which provides beautiful penetration depth but lacks in resolution. So photoacoustic imaging falls in this sweet spot where it could provide a three-dimensional image of the tumor at a resolution of about few hundred to few hundred microns at an enough penetration depth that we can use to look at the whole three-dimensional tumor volume and see the changes in oxygen saturation as with respect to the treatment. So going into the introduction of what photoacoustic image is, there are several review papers, but I try to put it in as simple terms as I can. Let's assume we have a tissue with an absorber such as a blood vessel that is pointed here. In photoacoustic imaging, you shine the tissue with a short laser pulse, a nanosecond laser pulse. The absorber takes in this energy, undergoes a thermoelastic expansion and contraction, which causes the generation of acoustic waves that are then detected by an ultrasound transducer. Given that one way is light and the detection is through sound, the same receiver electronics used for ultrasound imaging can be used for photoacoustic imaging as well. So naturally, you would get ultrasound and photoacoustic images that are spatially co-registered. What you see here is a representative photoacoustic image of the tissue. Since the tissue in the surrounding environment is scattering and has no absorbers, we do not see any photoacoustic signal, versus the absorber clearly gives an enhanced photoacoustic signature. So since the photoacoustic signature is dependent upon the optical absorption properties, oxy and deoxyhemoglobin have different optical absorption properties. And if we do spectroscopic imaging, or that is imaging at different wavelengths, we will be able to tease out the oxygenated and deoxygenated hemoglobin percentage, or the oxygen saturation within that particular blood vessel. So with this in background, I'm going to skip the electronics and image processing part of it and directly go into validation, or show you the images of how photoacoustic oxygen saturation maps look like for a subcutaneous tumor. So what you see here is ultrasound image of a subcutaneous tumor. As I mentioned before, ultrasound and photoacoustic images are inherently spatially co-registered. So, and with the Vivo laser system, we can obtain both these images simultaneously. So here we see the H&E image on the right-hand side. Obviously, we can see the structure, but we have no information on the oxygen saturation of the blood vessels in this tumor. So across the same cross-section, you see a oxygen saturation image below, where blue represents low or hypoxic regions and red represents highly oxygenated regions within the tumor. So if we look at this image carefully, you notice that there is a highly hypoxic region in the top left corner, and the bottom part of the tumor is oxygenated. That is, the blood vessels are feeding this tumor from the bottom up, and probably on the side there are a few more blood vessels here. So we euthanize the mice at this point and try to get a cross-section for histology across a plane that is similar to what you see in the images. And here's the immunofluorescence image for hypoxia markers. What you see here is CD31, which is a biomarker for blood vessels, shown in green. And pimenidazole is a hypoxia marker that stains all the regions that have a PO2 less than 10 mmHg. So again, if you compare the immunofluorescence with the oxygen saturation image, you see that the top left part of the tumor that I'm pointing to here is very hypoxic, which is what this region is. And the lower part of the tumor is highly oxygenated. Therefore, the pimenidazole doesn't stain in this part of the tumor. So there is a very good spatial correlation between the oxygen saturation maps and the immunofluorescence. So having validated it, we wanted to now go ahead and do our therapies and longitudinally look at the changes in oxygen saturation with therapy. So again, going back to the question, two questions. We wanted to identify which tumors are responding to the treatment, which are not, and if they're not responding to, if they're not responding to the treatment or only partially responded to the treatment, can we predict the tumor relapse at an early time point? So for that, we focused on vascular targeted photodynamic therapy as a model therapy treatment. But this particular technique that I'll be presenting can be applicable to radiotherapy or any other anti-angiogenic therapies as well. So little bit details on photodynamic therapy. As I mentioned, it is a photochemical modality where we have a photo sensitizer and a light, and both are needed for the photodynamic action to happen. The light excites the photosensitizer to a triplet state that reacts with the ground state oxygen to cause reactive oxygen species that cause the cell death. So if the photosensitizer is localized to the blood vessels, the hypothesis is that the blood vessels are damaged, which will cause the change in oxygen saturation. And through observing the changes in oxygen saturation, we can predict tumor destruction or non-responsiveness of the tumor. So going into the experimental details, we did these experiments on subcutaneous tumors with U87 glioma cell line. The tumors were about 6 to 8 millimeter in diameter. We used an FDA-approved photosensitizer, benzoporphyrin derivative, and it has an absorption at 690 nanometers. So we shine the light on the tumor at a particular dose. In this case, we used 100 joules per centimeter square. And another important thing to note is the drug light interval. Here we used 1 hour and 3 hour drug light interval. The drug light interval is the time from when we inject the drug to the initiation of the light. And it is important because if the drug light interval is too long, then the photosensitizer is out of the tumor, and even though you shine the light, there is no interaction between the photosensitizer and light to cause the PDT action. So that also the drug light interval plays an important role in determining photodynamic therapy efficacy. And we image these tumors before doing PDT and 24 hours after doing PDT. So as you can see here, in some of the mice, we did get complete response. And some of the mice where the drug light interval was very long, they did not respond to the PDT action. So we went and looked at the photoacoustic images. So what you see here is a pre-PDT image of a tumor that completely responded to the treatment. This image is an overlay of ultrasound on the background. The grayscale image that you see on the background is ultrasound image from where we identified the tumor region and circled in green. And we overlaid the oxygen saturation map on top of that, on top of the ultrasound image. So again, here red represents highly oxygenated regions and blue represents hypoxic or low oxygenated regions. And where there is no signal, those regions are avascular regions. So in complete responders, what we see is 24 hours post-treatment, the tumor really gets hypoxic. That is, what is happening is the blood vessels are damaged, so there's no exchange of oxygen. So the tumors are, so there's no oxygen saturation signature there. So we observe that the tumor is completely hypoxic, or has though it has blood vessel signature, that is, there's hemoglobin, it is all deoxyhemoglobin. And in non-responders, prior to PDT and 24 hours PDT, we do not see any difference in oxygen saturation in these tumors. So we quantified these in several mice and looked at the changes in oxygen saturation. So in complete responders, the oxygen saturation reduces to less than 10%, and this data is highly significant, versus in non-responders, there is no change in oxygen saturation before PDT or 24 hours post-PDT. So this is all great. So it's all quantification of the data. But the real question comes in is, can we identify treatment responders? So the idea was, yes, we can. If the oxygen saturation is reducing to less than 10%, we can say that a particular tumor is responding. And if not, then the tumor is not responding. But then the second question is, can we predict the tumor recurrence? What is really happening in the tumors which are represented by this red line here? That is, they sort of seem to be responding for a while, but then regrow back. So in that case, what we did was we took the advantage of three-dimensional imaging of ultrasound and photoacoustic imaging. So in all the mice, we actually did a three-dimensional scan. That is, what you see here is 2D B scans across the tumor volume. And the ultrasound image gave us the tumor anatomy. It gave us the 3D tumor volume. And the photoacoustic oxygen map can be overlaid on top of this 3D tumor volume to exactly see, which areas are highly oxygenated, which are not. And if we have this three-dimensional data at different time points and put it through an algorithm which says which of these regions or which voxel actually changed the oxygen saturation to less than 10% post-therapy, we can compute a treatment prediction map. So, which is what we did, and through this algorithm, we said if a particular region changes the oxygen saturation to less than 10%, say that region is treated and color it in green. And if there is no change in oxygen saturation, allocate it to the no response to treatment voxel and color it as red. So we're trying to make this complicated, color map into a simple color map as green and red to sort of come up with a prediction map that can tell us which regions of the tumor are treated and which are not. So I'm going to show you a few comparisons with photographs of the tumors that were treated, and we'll be looking at the top view of the prediction map so that we can compare it directly with the photographs. So I have three columns here, the before PDT and post-treatment photographs, and the prediction map is shown in the center column. So this is an example of a tumor that did not respond to the treatment. So you see before PDT and after PDT, the tumor just keeps growing, and the prediction map shows that the prediction map shows that it is in red, which means that the oxygen saturation did not change in this tumor before and after treatment. And then this is where things get interesting, is these two tumors were treated the same way. They had the same drug light interval, same light dose, same photosensitizer dose. They're implanted with the same cell line and concentration of cells. And then 3 days post-treatment, you can actually see necrosis in these tumor regions. The whole tumor seems like it was treated. But then what happened was 23 days post-treatment, the top tumor you can see is completely gone, and the prediction map actually shows that the oxygen saturation reduced to less than 10% and that it is a responder. But in this last panel, you do see that a particular region of the tumor the oxygen saturation did not reduce compared to the rest of the tumor. And I'll go over why that happened, but it's fascinating to see the correlation between the photographs. It's exactly this top region that actually regrew back in this mouse, and then it became into this nasty tumor within a week of treatment. So using the prediction map within 24 hours post-treatment, we were able to say that this top part of the tumor was not successfully treated. Probably the dose was not enough to treat that part of the tumor, and that could actually cause recurrence in these tumors. So there's another interesting example. We wanted to validate again, how good this prediction algorithm is. So what we did was, this is the tumor. We covered half of the tumor in a black cloth so that, we stopped the light from getting to the tumor, and then we did the PDT illumination. So ideally, we would expect only the top part of the tumor to be treated. The bottom part of the tumor, because of this black cloth, the light wouldn't get in, so there would not be any PDT action happening in the lower part. And from the photographs, you can actually clearly see the top part getting treated, and the bottom part of the tumor is not treated. But then when we look at the treatment prediction map, what we see is we identified 3 regions. So we do identify clearly the region that was not treated due to the black cloth. But in the top part, we saw 2 regions. One is a treated region, so the center part got treated, but clearly the top part of the tumor did not respond well to the treatment. That is, the oxygen saturation really did not go down to less than 10%. And exactly from that top region is where the regrowth starts occurring, and then 10 days post, it becomes a nasty tumor again. So we try to validate this with caliper treatment. So in the next slide, I'm going to show you that, a graph of correlation. So on the x-axis, we have the axial length that we measure from the photoacoustic prediction maps that were obtained 24 hours post-therapy. So these are the axial length of the red regions calculated from the prediction map. And on the y-axis, this is the caliper measurements of the axial length, again, of the regrowth tumor or the recurring tumor 20 days post-therapy. So what we see is a good linear correlation where which indicates that higher the higher axial length that the prediction map shows, the greater the tumor volume, or the axial length, or the greater the tumor volume. So going back into the question as to why there are a few regions within the tumor that did not respond when we actually shine the light over the whole tumor region, is we went back and looked at the total hemoglobin within different regions. And what we noticed is these regions, these regrowth regions actually had higher concentration, or higher density of blood vessels. So the dose that we were giving was not sufficient to damage all the blood vessels. So there were still some viable blood vessels remaining in the region that actually caused the tumor to regrow back at a later time point. So now our ongoing studies are to treat this, identify this region within 24 hours, treat this, and make sure that the tumor does not regrow back again. So this work is published in Theranostics, and we made it to the cover feature, so that's great. So in summary, using 3-dimensional ultrasound and photoacoustic imaging, we were able to identify treatment responders, particularly looking at oxygen saturation 24 hours post-therapy, post-PDT. We were able to identify which tumors are responding, which tumors are not, and then look at the 3-dimensional oxygen saturation maps and come up with prediction maps that can exactly point out to where there was not enough dose reaching the tumors, where the treatment was not effective, and identify the regions that could regrow back post-treatment. So our future work is looking at other therapies. We are looking at different anti-angiogenic therapies, small molecule therapies, and to to see if this particular technique holds, and if the changes in oxygen saturation could be used to monitor, to predict recurrence. And we're also looking at the possibility of monitoring PDT online and adjust the dosimetry accordingly using, again, photoacoustics for customized treatment. So with that, I would like to end the talk, and thank you very much for the opportunity, and I'll open it up for questions.
Great, thanks very much, Dr. Mallidi. That was a great presentation, really sort of highlighting how, photoacoustics in general and ultrasound as well can be used to sort of monitor these different things like tumor volumes and treatments and that kind of thing. We've got a couple of questions. I'm actually going to leave the sort of presentation with you so you can flip back and forth in your slides if you want to show anything again. But there are a couple of questions here. There's one coming in, is the identification of tumor regions as darker or low ultrasound signal regions on ultrasound images automatic or manual? Yeah, so they're asking, is the identification of the sort of B mode, just the ultrasound, is that an automatic or manual thing? Those, I think they're referring to those ROIs that are drawn there.
Okay, yeah, so it is a manual thing. So there, with some experience, we can identify the tumor regions, especially in it gets difficult in orthotopic tumors, but is doable. It is very easily detectable in subcutaneous tumors.
Okay, great. Yeah, I think they're getting at, they're sort of getting at, you see this tumor with your ultrasound, but how do you know where the tumor boundaries are and that kind of thing? Do you mind just making a comment about that?
Sure. So I'll talk in perspective of this particular data that I show here. So these are orthotopic pancreatic tumors, and I do agree that for the tumor boundaries get difficult to identify sometimes, especially if there is a lot of shadowing effects, things like that from the intestines and the food in the stomach. So we actually try to fast these mice for a little bit so that those effects are gone, those artifacts are not there. And usually the tumors are hypoechoic, and they have a very different ultrasound signature compared to, let's say, kidneys, which are in the case of pancreatic tumors. So the boundaries are fairly easily identifiable in case of surgically implanted tumors, that is, if you implant cells in the pancreas. Things get a little bit difficult with transgenic models, but after the tumors grow to a certain size, let's say 2 millimeters or above, it is easy to identify the regions.
Okay, thanks. I've got kind of a related question. It's sort of just getting at that kind of thing, not just in the ultrasound, but the photoacoustics as well. I just wanted to ask how important to you is resolution in these kind of studies?
So obviously, photoacoustic falls, in sort of in the mid-range, and we really cannot, look at cellular resolution with photoacoustics, but what is helping us is trying to get the three-dimensional perspective of the tumor at reasonable resolution, and identifying the regions, where the blood vessels did not shut down. I think, where we don't need cellular resolution as long as we have, say, about 500 micron resolution, and be able to see that in perspective of the whole tumor volume for designing therapies and monitoring therapies. I believe that should be okay for preclinical studies.
Okay. Okay. I mean, I'm sort of noticing that these tumors are on the order of, half centimeter to a centimeter, and you're looking at, sub-regions, in this tumor. And another somewhat related question is someone's asking about the prediction map. So when you're looking at a prediction map of the completely treated, the green regions, versus the uncompletely treated, say the red regions, the person's asking, is the boundary between these different regions, is it really clear and continuous, or is it possible that you've got, let's say, say, a green region which is surrounded by a large red region, or vice versa? Or is it usually fairly continuous?
Because, I agree with that person asking the question. So as you see in the map here, it seems like as if it's a clear-cut demarcation, but actually it's not. One thing to make sure is here I used a particular threshold, and it is because of this threshold that we do see, strong, treated region and non-treated region. But there are in some tumors where we see, especially at the bottom, in subcutaneous tumors, there are a lot of blood vessels feeding from the bottom of the tumor. So there's this top region that's completely treated, and the bottom part of the tumor is not treated. So we do see images like that too. It again depends upon how you are thresholding your algorithm. In case of PDT, what we noticed is less than 10% a change, less than 10% oxygen saturation gave us good correlation with the caliper measurements, but that could change depending upon what treatments we are using.
I see. Okay, so if you change that threshold, you might actually see more sort of heterogeneity in these prediction maps, possibly.
Yes.
Okay. But you chose that threshold because it actually
again, from our 2D, actually, we chose that from this number here. So we did several mice, and then from the complete responders, our mean was about 10%. So we chose that as our threshold level. But depending upon your therapy, your response, it might be different what your threshold levels are.
Okay, great. I've got a couple of more questions. I guess these were all done on sub-Q models. At least the PDT portion of things were done on these sub-Q models. How do you see this working in an orthotopic model in these animals?
Sure. So those are the studies that are ongoing right now in our lab. We are looking at changes in oxygen saturation in orthotopic pancreatic tumor models. So obviously, again, the penetration, light penetration effects do become a problem in certain cases, and we are trying to use different like focused light beams and other things with the help of VisualSonics, and also use contrast agents to help identify the tumor regions.
Okay, great. And I mean, what's the sort of, just in general, the status of PDT in the clinic, or how do you see these sort of treatments and maybe treatment monitoring sometime in the future, and the technology being used to do this kind of thing in the clinic?
Sure. Right now, PDT is used for acne treatments and is in phase 2 clinical trials for pancreatic and several other oncological applications.
Okay.
So right now, fluorescence is primarily used. So we look at the amount of photosensitizer or drug in that particular lesion, and then design dosimetry. But PDT action is so complicated that just looking at fluorescence wouldn't suffice. We need to also monitor oxygen, or the oxygen content, which plays an important role in PDT action. So by monitoring oxygen saturation with something like photoacoustics, you would be able to customize doses for that particular patient.
Okay, thanks. I'm just going to give you one or two more, just sort of a practical question. I noticed that you have this really nice co-registration between the histology and your ultrasound and photoacoustic images, and I know there are entire research programs dedicated to doing this kind of co-registration between modalities. But how did you do that in this case?
So what we did was, this is work of several hours and several days actually. So what we did was we took the tumor out post-euthanasia. and embedded it in the same orientation as the imaging in an OCT mold, and we actually sectioned the whole tumor and stained all the cross sections. We sectioned about 150 micron apart, which is what the three-dimensional, which was what the step size was in 3D imaging, and then went back to our images and took our histology images, and then went, one after another to see which one correlates well with our images, approximating, the distances that we have from the histology and also from the 3D images.
Okay, yeah, it's really nice work. Those images look great, and it's really nice to make that comparison, the in vivo comparison with the sort of ex vivo, which I think a lot of people in cancer research are used to that, ex vivo element, and seeing it in vivo is really nice.
Sure, thank you.
Okay. I think we'll end it there. I just want to thanks everyone for attending. Thank you especially to Dr. Mallidi for presenting this really exciting work. I just want to show a couple more slides. We're going to be, we've got some upcoming webinars. Yeah, we've covered the questions. Yeah, and Dr. Mallidi mentioned this presentation on the cover of Theranostics. It's a great paper. If you want all the details, obviously, look up this paper. It's in the March 2015 issue of Theranostics. You can get all her sort of materials and methods and that kind of thing there. And of course, if you want, you can send us questions, and we can pass them along and connect you with Dr. Mallidi so she